Does a Boston University (BU) study of a COVID-19 strain with an 80% lethality have implications for humans? No, that's not true. The kill rate refers to mice used in the lab tests, and BU's manipulated virus was less lethal than existing wild viruses, which showed a kill rate of 100%. The experiment conducted at BU manipulated the SARS-CoV-2 Omicron virus variant and tested it against two other strains to determine what mechanisms made Omicron more infectious. The various forms of COVID were compared through testing on tissue and mouse models, not humans. Experts say such studies are common in defining the characteristics of a virus. The claim emerged in an article published on October 17, 2022 by the Daily Mail, and then other media including Fox News, that claimed researchers at BU created a new deadly COVID strain with an 80% kill rate. Here is how the Daily Mail post read at the time of this writing, quote, "Exclusive. This is playing with fire. It could spark a lab-generated pandemic. Experts slam Boston lab where scientists have created a new deadly COVID strain with an 80% kill rate." End quote. This is how the article appeared at the time of writing. Source: Daily Mail. Screenshot taken on Friday, October 21, 18 hours 18 minutes and 26 seconds 2022 UTC. Research using live and highly infectious viruses can theoretically start a pandemic if safety equipment and practices fail. BU's research was correctly carried out in a lab with the greatest safety level, level 4, and critics have not documented any failures in the facility nor in the conduct of the study. The issue critics have raised is whether BU correctly interpreted federal rules on notification to the National Institutes of Health (NIH) and local rules of the same nature. The Daily Mail article claimed that the research team made a hybrid virus, combining Omicron and the original Wuhan strain, that killed 80% of mice in a study. While this is technically true, the study is not applicable to humans and was conducted in the Level 4 BU lab. Furthermore, this type of research is common in determining what elements make a virus infectious or cause severe infection. In a public statement, BU researchers said that the Daily Mail reporting was false and inaccurate, and that this research made the virus replicate less dangerous. The study is published in the preprint server bioRxiv. It has not yet been peer-reviewed in a scientific journal. Scientists often publish early findings in preprint servers to seek input and speed the development of new research methodologies, particularly throughout the course of the COVID pandemic. Study author Ronald B. Corley, microbiologist and director of the BU National Emerging Infectious Diseases Laboratory, NEDL, said that the Daily Mail pulled one line of the paper's abstract out of context: deadly COVID strain with an 80% kill rate. Even so, the study sparked concern in the scientific community over whether the BU team received the necessary approvals to conduct the research in the first place. A chimera virus to determine pathogenicity. To determine how Omicron, a coronavirus strain that first appeared in 2021, is able to evade immunity, scientists at NEDL created a chimera virus by extracting Omicron's spike S protein. The spike protein is the structure that allows the virus to invade human cells, making coronavirus particularly infectious. Researchers attached the S protein to the original wild-type strain to determine if that increased the original virus' ability to infect mice. This chimera virus was then compared against the unaltered original wild variant. The chimera was shown to cause 80% fatality, 0.8, in mice, which is less than the 100% mortality, 6/6, caused by the wild variant. These findings signaled that, while the spike protein is responsible for infectivity, changes to other parts of the virus structure determine its deadliness. In other words, the spike protein is not the primary determinant of Omicron's viral pathogenicity, or what the National Library of Medicine (NLM) describes as the process by which an infection leads to disease. BU's approach was only tested on mice. Consistent with studies published by others, this work shows that it is not the spike protein that drives Omicron pathogenicity, but instead other viral proteins. Determination of those proteins will lead to better diagnostics and disease management strategies, lead author Mohsen Saeed said in a statement emailed to Lead Stories on October 19, 2022. Did the study receive the necessary approvals before being conducted? The research was reviewed and approved by the Institutional Biosafety Committee at BU, as well as scientists and community members at the Boston Public Health Commission.